Okay, well, so this is Stefan. So he's kind of been pioneering a lot of these, a lot of the work in terms of like how promoters interact, or like the proximity between enhancers and promoters. So a lot of that technology, Stefan has been doing a lot of work on that, and recently has been doing a lot on pluripotent cell states and how things change between states, which is kind of what that picture is. But there's a lot of work to be done here, I guess, in terms of like how you analyze this stuff. But I thought actually this stuff, the analysis was kind of cool. It'd be interesting to see what people from a different kind of physics perspective would think about the way you actually think about something which is not really one-to-one necessarily interaction-wise. So I think that's kind of be interesting to see what Stefan's done since. And yeah, it'd be great to see if there's any way that I can be better ways to model this kind of stuff, because I also struggle a lot, kind of follow whatever Stefan does. So
I struggle too. Yeah. Thanks for the invitation. And I've been told this is kind of an informal meeting. That's great. Interrupt me whenever you feel like it. So as Sunjan said, I was going to talk about cis-regulatory control of pluripotent stem cells. Taking a step back, the question that has really fascinated me for the last 10 years or so, 15 really, is this one: how remote enhancers relay regulatory information to their target promoters, and which has been called one of the central mysteries of genome function and organization. And so I want to talk, this is the kind of the structure of my talk. I'll start with a short introduction on enhancers, then I mention the technology that we've developed, as Sunjan said, to capture enhancer promoter context genome-wide. And then these next two are kind of whistle-stop tours. Cursor in the middle. It's not mine. Anyway, ignore. And so this is published work, largely published work, but I wanted to highlight some of the findings that we gained from applying this methodology. And then the next two are unpublished projects that are ongoing in the lab, which I'd like to share with you. So I'll start by introducing enhancers to you. Most of you will be familiar with this, but enhancers are really very cell type specific gene regulatory elements. That's this big here, so red, whereas gene promoters, genes are largely located in the same genomic positions. Enhancers are not, they're very cell type specific. And we have means to localize enhancers genome-wide, mainly chip-seq methods like that. I'll come to that later. They're imperfect, I think, but we can map enhancers genome-wide. But what's harder is to know then which target gene they regulate. For example, this is a famous example, the lymphoid specific enhancer of the spoiling the surprise here. So, for example, this is an enhancer that sits in the intron of the LMBR1 gene. So if you didn't know anything about this, you would probably assume it regulates one of the 4 genes in its proximity. But it doesn't. The enhancer it regulates is actually almost 1 million base pairs away. And it encodes the developmental regulator sonic hetero. And it does so, that's the prevalent model, at least at the moment, by forming direct physical contacts with the target gene promoter, with the intervening DNA looping out. So what happens if you delete this enhancer experimentally? You can see that the mouse are born without limbs, or with severely truncated limbs, so limbs are fine. And this really demonstrates the cell type specificity of these enhancers, because sonic hedgehog is very important in many developmental pathways, but its expression in other organs is absolutely fine. It's specifically in the developing limb that this enhancer is crucial, and that's why this pathway is disrupted. And interestingly, point mutations in the enhancer in humans, in different patients, lead to polydactyly developmental malformations. So the points I wanted to make here is that the enhancers are cell type specific gene regulatory elements, key regulators of cell fate, and they convey regulatory information through chromatin loops. This is a beautiful experiment that think, a milestone experiment that convinced me to work in this field, really. And that is an assay called forced chromatin looping. So what Global's lab has done in this paper is they've constructed artificial zinc fingers that would bind to the LCR, this is the beta globin super enhancer, and to the beta globin gene, and both were fused to a protein called LDB1 that homodimerizes. So what it does is kind of, it acts as a tether to bring the enhancer together with the promoter. You can actually substitute LDB1 here with anything that homodimerizes. It's a tethering. And this activates beta globin transcription over 1,000-fold. So inducing mere proximity between enhancer and promoter can really activate transcription quite dramatically. Doesn't always. I didn't know that back then. I thought it was simpler than this, but biology isn't simple. Oh, that's just an intervening sequence that I've made to mark that. It could be another globin gene. So the globin gene cluster is, but it's just for visual. That's about 50 kb, which was back in the days was considered long range. No, it's shifted. So next I wanted to talk about the technology we've developed. And this is based on Hi-C, which some of you might have heard of. So Hi-C is a technique that captures, in theory, all interactions between every region of the genome with every other region. So how it works briefly is you fix chromatin together, and by that then you cut with a restriction enzyme, and then you this is a bit of a detail you put in biotin, but then you ligate. So the key steps are cross-link, cut, and ligate. And what you do is then you've generated this hybrid molecule. So cross-link together, cut and ligate. You've generated a hybrid molecule that doesn't exist in nature, but that you can read out by next generation sequencing. And the frequency at which you detect this is a measure on how often these sequences were close enough in proximity that event could happen. So it's a really powerful method, but its power is its weakness at the same time, because they're incredibly complex Hi-C libraries. In the beginning, that brought them a bit of a bad reputation, because people saying, well, it's not really reproducible. And it's not reproducible because it's an undersampling issue. You can actually sequence the same Hi-C library twice, and it won't be reproducible. We've learned this the hard way. So without that can't be technical, right? And that is because if they're so complex, you will sequence the tip of the iceberg, and a slightly different tip of the iceberg, you do it next time. So this number is, even though next generation sequencing is incredibly powerful these days, the complexity of Hi-C libraries still dwarfs it. And actually, this is kind of based on calculation with a 6 base pair cutter. If you use a 4 base pair cutter, you have 100 times more complex libraries. So it gets even worse. So when we realized this, we thought, well, we can develop an assay maybe that zooms in on the parts of this Hi-C library that we're most interested in. And for us, we wanted to understand promoter-enhancer interactions. We thought we'll start with promoters. So what we did is we designed a library. We designed it, and Agilent made it for us, a library of biotinylated RNA baits. It's a bit like exon capture, if some of you are familiar with that. Biotinylated RNA baits that targeted all gene promoters in the human or mouse genome, so 22,000 promoters or so. And if we incubate that with a Hi-C library, we can fish out, through biotin strip interaction, we can fish out the promoter interactions, right? So we did it for all promoters, and so we do it simultaneously. So for 22,000 promoters in the human genome. But they just put it down. You design sequences. You design RNA. So they're biotin RNA, biotinylated RNAs, 120 nucleotides long. And you just need to make sure they are specific. So normally what we do, you think of restriction fragments, we have one that's here and one that's here. So you have to think in restriction fragments when you work in this in high-C libraries. And sometimes we only have one because the sequence on one end is either repetitive or yeah, mostly because they're repetitive. But we do that, so we capture it in a single experiment for all promoters. And it's, I mean, we took a cutoff at some point, and I'm saying all. It's never complete, but it's a modular. You can easily change that. You know, the experimental flow doesn't change at all. We've actually done it against. You know, you could do it against CTCF, all CTCF set in the genome. You can do it against all enhancers and specific sets. Promoters was nice for us as a starting point because we can use the same system to profile different cell types, because largely the promoters are in the same place. Not always, and we'll miss some of those, but you know, for genome-wide trends, we're okay. So we did, yeah, and reduce the complexity of these high-C libraries, and that makes them much more amenable to sequencing. So I'm giving an example here. This is from a high-C library where every arc here is an interaction in a kind of gene core region around the cell 1 locus. In a high-C library, when you zoom in on the interactions that are over this promoter, there's much fewer, but more important, reproducible. That becomes with these sampling undersampling issue. When we use promoter capture high-C, we get much more interactions, and they're highly reproducible because we're sequencing. We've reduced the complexity of this library massively. And this is just one. This is just to show you we can do this consistently. So in blue, you always have in a high-C library, the reads that by chance fall over one of those promoters, it's always 5%, like 4.9, 5.1. It doesn't really change by cell type, as you would expect. And we enrich that. The theoretical maximum would be 20-fold. We enrich it by a factor of 15 on average. So it works well. So what have we gained? Oh, this is yeah. Sorry, yeah. Do you want me to go back? The previous, yeah.
8 times larger than the regular vaccine.
Well, we enrich for those. I mean, it's specific because we have a specific question. That's not to say that the 95% of these other interactions that are there are meaningless. You know, we're missing things like, for example, enhancer interactions, which could be really interesting. You can design the experiment in a way that would capture those. For us, in this application, we wanted to, we thought we'd find, you know, all the enhancers that regulate all genes. It wasn't that simple, but we've made good progress. It wasn't quite that simple, but yeah. So this was the first control that we did, so to convince ourselves that it had worked. What we've done here, we grouped the genes by their promoters by the expression of the associated genes. So from non-expressed to highly expressed here, and then we looked which histone modifications are enriched in the interacting fragment. Right? And what we were hoping to find is that the enhancer marker, this is H3K4 monomethylation, is kind of an inactive neutral enhancer marker. H3K27 acetylation is an active enhancer marker, that they would be overrepresented in the regions that interact with highly expressed genes, which is what we expect, where we expect enhancers to be, and that's indeed the case. Right? So we thought that was as proof of concept that was a relief. It's not that well known. I mean, it's also important to consider. I always thought, and some people still think that all of these interactions are with enhancers. Only a minority are. There's many other regions that we actually don't know what they are. So
it's systematically interacting with these regions very reproducible.
Yeah,
but they're not enhancers.
They're not enhancers. Yeah, no, I mean 25% for highly expressed genes, 25 to maybe 30%, depending on the experiment or so. In the interacting regions are enhancers. That means they're highly enriched. but the majority of the interacting regions, even for highly expressed genes, not enhancers. I found that troubling in the beginning, but I find it interesting now. But we don't really know about. I mean, we can make some predictions about some of them. You know, some are found by CTCF or so. So we can make predictions, but many of them we don't know. Yeah. And so then, what was nice about this, even though we had designed this technology and developed this technology to answer a question in basic biology, namely how enhancers interact with promoters, or which enhancers interact with which promoters genome-wide. What we realized very quickly is there was a big interest from the GWAS community in this, so genome-wide association studies. So basically, efforts, large-scale effort that match sequence variation to disease risk, disease susceptibility, disease risk. And when we look at, this is now 10 years old, but this is across all GWAS data sets that were available then, and the trend is still there. What I think people were expecting was to find that most of these regions would fall into protein coding sequences. And only 5% of them do. So that means they're overrepresented because they occupy a space in the genome of only 2%, so significant overrepresentation. But it leaves 95% of these regions outside of protein coding regions. So, and a lot of them look like enhancers. They're close to DNS1 hypersensitive sites. So our technology offers a means to assign these regions to their putative target genes across for disease states. I know little about disease and even less about GWAS. So we've founded a company that now takes care of this. It's in its third year now. We've grown to 25 or so now. So it's been very exciting to be part of that as well. Yeah, and I wanted to talk you next through a few insights that we gained from applying this technology. When we started, there were these two conflicting models in the field about enhancer-promoter interactions. The first was the de novo or instructive model that would predict that enhancer-promoter interactions form during or concomitantly with cell lineage specification. So they're absent in precursor cells, but in the progeny where the cell is then expressed, they're present. The other model predicted that they are already preformed in a precursor state, but something stops them from being active. And really the switch that switches a gene on doesn't correlate with the formation of a new promoter enhancer interaction, but could be a change in the enhancer state, for example. And we've looked, this is just one example slide, summarized a huge amount of data there. We've looked during cell lineage specification. So in this case, human ESC, embryonic stem cells into neuroectodermal precursor cells. And without going into detail, essentially what you can see during differentiation, wherever we've looked, you see both. You don't see one or the other. You see de novo formation, and you see pre-established contacts that then change enhancer status. So something might already interact with its target gene, but it goes from an inactive enhancer to an active enhancer, something like this. So we saw that consistently. It's not one or the other. You find both, and you find both abundantly. We also then ask what happens if we don't look at situations where really the cells change their state completely, but they react more short-term and often reversibly to a short-term stimulus. And for that, we teamed up with Tony Green's lab, and they had a system where they exposed HPC7 cells, the hematopoietic precursor cells, to thrombopoietin. That's an inducer of megakaryocyte receptor differentiation in vivo. And they've studied this system for years, and we knew that even a relatively short exposure of 30 minutes results in dramatic changes in gene expression, 2,000 genes change in expression, 10,000 differentially associated regions. So transcription and chromatin states change quite dramatically in this situation. How about 3D genome architecture? This is a bit like a spot the difference picture, because that's what it was. Nothing changes, essentially. So these are all preformed. Essentially, all this kind of the entire signaling landscape works through preformed interactions. That's what we found. That was very clear. Whereas in differentiation and cell chains, we see a difference of both. And this is consistent with other studies, TNF alpha, and what other people have found. So, so I wanted next talk about polycomb-mediated chromosomal interactions in mouse and even actually mouse in this talk only, pluripotent stem cells. And that's is one of my favorite stuff. Yeah.
When you see these kind of how easy is it to get a like subtle changes, like you get a like percentage change or like this complex a little bit like
yeah, but it's dangerous to set like tight thresholds on this. You know, we have to, for example, in the pipeline we use to call these interactions is called Chicago, as you know, and that has you know, a specific score from which it scores significant interactions, and that's okay. But if you compare two different cell states, so that's dangerous. You don't want to pick up something that's 4.9 versus 5.1 and say, oh, that's a differential interaction. So you have to have something more sophisticated there. So
yeah, we'll struggle with it. You get all these different scores, all of these different types of interactions, but I just say if there's a subtle change, how do you do it?
Yeah, I mean, I try to avoid this kind of, I take a bit naive view of that, but I try to avoid kind of hard thresholds for these kind of you know, I need to be more confident that I see a real change. So I would, for example, then ask that an interaction has a score of like random values, but over 5 in one condition and under 3 in another, something like this, that I'm not dealing with like minute changes. Yeah. So, it's a bit of poor man's vision approach to it, but I want to see stuff that I believe. So, and this study started, this is driven by Sarah Elderkin, started with the question we ask when we have promoter-promoter interactions, and which promoter capture as he fishes out as well, right? And what are the genes that are bound on both sides? Sorry, the proteins, of course, that are bound on both sides of this interaction. And when grouped up, we found that, to our surprise, it had been described that OCT4, NANOC, and SOX2 were major mediators of the 3D genome organization in mouse pluripotent stem cells. But we found that the highest ones, 3 out of the 4 highest ones, were actually members of the polycomb group family. So there's just 2 complexes. The top one is the catalytic subunit. Ring 1b is the catalytic subunit of complex called PSC1, polycomb repressive complex 1. So Z12 and EZH2 are members of the polycomb repressive complex 2. And Sarah had so we found that this in mouse embryonic stem cells, there was this network of very strong interactions, really long range interactions, sometimes trans interactions between different chromosomes that were centered around the 4 Hox clusters, which are, you know, the examples for polycomb regulated regions. They encode transcription factors that drive polyexis specification, these kind of things. So we want them repressed in pluripotent stem cells. And we found this. And Sarah had a system where we could inducibly take out ring 1b, and then it's not shown here, but the whole complex actually falls apart, PSC1. And what we found in this situation was that the network almost completely falls apart. So we could really link ring 1b to maintaining these interactions. And there's always in the polycomb field, there's a bit of sometimes a bit of a fight, PSC1 against PSC2. I don't want to take sides in this. PSC2 is clearly important. It's also mutated in many cancers. But we had a specific question to address here. So we had also cells where PSC2 was depleted, and that's a knockout of EED, which is another subunit of this complex, and you can see that the protein's gone. Also, the histone modifications that these complexes set, which for PSC2 is H3K27 trimethylation, for PSC1 is H2K119 ubiquitination. They're gone, so there's no redundancy in this system. We can study this. There is for For the insiders of you in this example, you have to knock out ring 1A as well. There is a parallel there, but we've done that. So that system allowed us to look at what happens in contexts between sites that are co-occupied by PRC1 and PRC2, or that are only occupied by PRC2. That's almost so too small to really work with, that are only occupied by PRC1 in the mouse simulation. You can see examples. The sites down here. So we had a pretty good idea by looking in the wild type situation that the PRC1, PRC2 co-occupied complexes interacted, sites that are co-occupied by these two complexes interact much more strongly than the PRC2 only. But we wanted to look into knockouts. And so this is first the PRC, the sites that are occupied by PRC1 and PRC2. We take out PRC2, contact frequency drops quite significantly. We think probably through mistargeting PRC1. If we take out PRC1, the drop is. What happens when we do this? We take out PRC1, nothing much happens. There shouldn't really happen anything because PRC1 is not bound here. But surprisingly, with PRC2 knockout, nothing happens. So while this is by no means saying that PRC2 is not important, but the complex that drives long-range interactions in mouse embryonic stem cells is PRC1. So what happens to the genes in this spatial network? So we looked at in the knockout at the genes that are expressed. You can see that more genes, polycomplex, the repressor complex, more genes go up than go down. And when we compared all ring1B bound genes, of course, more go up and go down. Interestingly, and I can't offer like really an explanation for that, in this Hox network, so this network of these 8 genes centered around the spatial network, around the Hox clusters, the genes go up even more. So it could be that, but I don't know, it could be that they're not only repressed, they're also somehow primed for being really efficiently expressed later when the time comes. But we kind of a bit of a that's not development just interesting. So this was my favorite. What I've shown you so far is we talked about these promoters, really long-range interactions between. Of course, promoters don't come in, in isolation. They bring their own network with them, so they bring enhancer networks with them. So what happens to them? What we saw is that they're kind of poised enhancers when they come in with these sort of poised. And what we found is that the interactions maintained between different enhancers, which is the opposite of what I would predict, because I thought the promoter network falls apart, surely the enhancer network also falls apart. But the enhancers remain associated with the promoter. They change their character. So they lose their recursive almost all of them, and they gain HDK27 acetylation mark. So it's a switch from a repressive to an active chromatin mark, rather than a loss or gain of chromatin interactions in this. We don't know. Yeah, it's a good question, but yeah, I mean, we'd have to. I don't know. Sorry, that's one.
Well, we are subject to hot themes. So there's this whole story about the linear organization of hot genes in the chromosome.
Yeah.
In the same order.
Yeah.
So does this in any way explain that correlation?
I don't think we can go that far. We're studying it in a mouse embryonic stem cell system here. So I don't think it'd be fascinating to study in a developmental context how how these genes are released out of this network, if they exist in this network. Is there sequential and temporal order how they are released? We haven't done that. So this was in a stem cell model. So I want to talk to you about cis. Yeah, sorry, more questions. We don't know. So it's been shown by Rob Close's lab that the catalytic activity is important for this. So he's made knockouts that the protein is still there, still expressed, still part of the complex, but it can't catalyze anymore. So that would implicate the modification in this. Yeah. So this is now a jump. This is some of what I've shown you. Actually, not the PRC2 part, because kind of we haven't published that yet. But the rest of this was published. I'm moving on to a study that's not published yet. So we were interested in enhancer variation, genetic variation, and how that shapes pluripotent stem cells and pluripotent stem cell potential. And we study this in human induced pluripotent stem cells, so that obtained from fibroblast and and so in collaboration with in Göttingen lab, who study primordial germ cells, sort of precursor against differentiation. And as a control, we use definitive endoderm specification. And what's fascinating is that we can find, and we didn't have to look very far. We have a panel of 10 iPSC lines that we work with routinely in the lab. We find that some of these lines are really effective, like I call them donor 1 here line, really effective in making primordial germ cells. These are primordial germ cell-like cells. Not relevant to this talk. Some are very, very ineffective. So you see that these are markers for the PGC. And the ones that, interestingly for us, the ones that are effective in PGCLC formation are very poor in making definitive endoderm, and the other way around. So we really found this kind of mirror image, that the ones that make PGCLCs quite suddenly very poor, and the other way around. So they have an intrinsic, they're not just kind of poor cell lines that you can't differentiate. They have kind of an intrinsic bias to go one way or the other, even though they share during this differentiation pathway, they share a common precursor. So we're now doing single cell multi-ohm and things like this to find out if we can link this to specific enhancer and genes. But it's interesting. And even one of the marker genes we choose is the same, SOC17. It's interesting that they have this mirror image. So what we decided to look, can we actually find differences already here that maybe would predict this kind of potential? And for this, we focused on genes that are quite highly. Most genes, I mean, these are all human iPSCs. Most genes are expressed at very similar levels. But there is about 150 or so that are quite dramatically differentially expressed between cell lines. So we looked at those. You can see an example of this, FOXC1, which is expressed during kidney development, normally should be silent in iPSCs, but in some cases it is expressed. We look at the ataxic profile across the locus in 4 cells. I'm showing you 4 or 10 here, but it gets too busy with 10. We look at the ataxic profiles, not much happening. That really surprised us. That still surprises me. I would have predicted ataxic maps accessible chromatin. I would have predicted there to be differences. When you see basically this kind of, this is the genes that we focused on have at least a 10-fold difference in expression. So quite dramatic. I was expecting that. But it made sense when Ferry, who was a visiting student in my lab, when she profiled chromatin marks across the locus. And suddenly we saw that, actually the next picture is even more convincing. We saw that wherever it's expressed, we see this. These are 2 bivalent chromatin marks. Martin knows a lot more about this than me. He's been studying this for a long time. But these are thought to keep these developmental regulators in a bivalent state. Sorry. My daughter. Need to check that later. She should be in school. She'll be all right. Yeah. Okay, just give me a second. We should be in school.
Hello. Hello. Why are you in school? Right.
Yeah. She always tells me she's not allowed to use her phone in school. Sorry. Is that recorded? Yeah. Can you edit that out? Yeah. Yeah, it is recorded. Yeah, but there's a maybe you can edit this. Oh, maybe not. I'll leave that to you. But actually, these marks are kind of there are a mix between an active mark and a repressive mark, and they keep these genes in a poised state so that they can be activated. What we see here is that balance tips, that you see a predominance of the active mark and the loss of the repressive in the line. Right, and this is even more kind of black and white. I think GBX2, an example expressed during neuron development. Again, you can see very little changes in the attack-seq, still something that I'm struggling to understand, but the balance essentially shifts when these genes get expressed. You see a gain of active chromatin marks and loss of repressive chromatin marks. So there's an erosion of polycomb group silencing, and we are now trying to understand the extent to which this contributes to heterogeneity between iPSCs and how much it could predispose them to, differentiating in one lineage or another. And this is the final story I want to talk to you about: functional enhancer annotation in human iPSCs. So I've talked a lot about enhancers already, so I'm coming kind of late with a slide that says what defines an enhancer, but it's a bit of a historic overview. So enhancers were originally identified by the ability to drive tissue-specific gene expression. So you use typically assays where reported gene is driven by a minimal promoter that doesn't drive expression by itself, but when you add an enhancer in front of it, actually the orientation doesn't matter. Suddenly you see a tissue-specific expression like this in the forebrain. And these days, this is the gold standard for enhancer still, but it's of course not really high throughput. ChIP-seq has largely replaced it for that reason, and the main marks we're using is CK27 acetylation, which marks active enhancers. That's the textbook. Some people also use attack-seq, which is a bit problematic because attack-seq only maps anything that's bound on chromatin. Doesn't really tell you anything about the nature of what's bound there. But when you look at in the literature in a bit more detail, with the enhancer browser, which is essentially a large collection of this kind of data, so reported in assays in mouse, you find that 40% of the tested enhancers, which have all the hallmarks of enhancers, fail to drive reported gene expression. So it kind of, and in an assay, a reported gene assay from Ian Chambers' lab, you found that only 11% of these regions actually drive reported gene expression. So it indicates that maybe if we rely on these approaches to map active enhancers, we are over predicting enhancers. On the other hand, there's also reports that find that regions that lack this enhancer mark can actually function as a, right? So these findings would suggest the opposite, that actually we're also under predicting enhancers based on this approach. So it's a mix of both, and we might not be profiling or looking at the correct ensemble of enhancers. So we decided to study this more systematically, and in human-induced pluripotent stem cells, we turned to an assay called STAR-seq by Alexander Stark's lab, and it's a very simple, elegant assay. So where you clone your regions of interest downstream of a minimal promoter and report it upstream of a polymerization site. So if this is an enhancer, it kind of drives its own, this enhancer acting in a position independent manner, it drives its own expression. So you can read this out in the RNA library. So the abundance at which you find these molecules is a readout for the enhancer strengths. a very elegant assay, you have to know, as with anything, its limitations, and I'll come to that in a moment. So we combine that with chromatin with other functional genomics approaches like a taxi, promote capture assay, which I've talked about, and cut and tag, which is a replacement for ChIP-seq in essence. And why is this important? So I'm showing you examples here. So we generated 3 StarSeq replicates in human iPSCs, each transfect 200 million cells or so. For a small lab like mine, that broke us almost financially and emotionally, but we got there in the end. And so the other reason why I'm showing this is so it's nicely reproducible. What you look at is always the enrichment of these tracks over this, which is our input. The green is our input. And I'm also showing this to alert you to the fact that not all of these regions, actually, majority of them do not overlap with ataxic signals. So they're not actually an open chromatin. So that's an important limitation of StarSeq. And this is shown here that in numbers, that what it measures is the ability of a sequence to function as enhancer in that cell type. It doesn't tell you anything about the chromatin state of that enhancer. So we focus ourselves on the ones that overlap with open chromatin. It's safer for now. I can't exclude the other ones, or at least some of them are enhancers, but the safer hypothesis for us is now that they have the ability to function as enhancers, but they're repressed by the chromatin state in that cell type and probably become active during cell lineage specification. So when we combine this we integrate this data with chromatin marks, we have this what I call chromatin repressed. So these are the ones that I just talked about. There are taxic negatives, so let's ignore those for a moment. We have these classical categories active, where you have H3K27 acetylation, the neutral or inactive enhancer mark H3K4 monomethylation, have an absence of the active mark H3K27 acetylation, and then you have the poised ones. So these are the polycomb regulated ones, where you have H3K27 trimethylation, which is the PSC2 mark that we talked about earlier. So our focus was for this study was on these ones, because I wondered, is it, they are thought to be primed enhancers, so to not be active enhancers in that cell type, but to prime genes for later expression during cell lineage specification. So we thought, well, is this, and this is just to show you example. I've not just picked an example that I like. These are genome-wide trends, really. So we thought, if this is a primed enhancer, is that the only possibility that because they drive reporter gene expression in that cell type, and they're bound by something, they're an open chromatin. So what's to say they're not active? So we considered this other model as well, that they might act as non-canonical, right? So they lack the active enhancer mark, but actually function as enhancers. And one of the assumptions you would make from the primed enhancer model is that the gene expression of the interacting promoters or interacting genes is low, or they're not expressed, right? They're only primed to be expressed later during differentiation. And when we look at that, we see that for the primed enhancers, the gene expression level of the interacting genes is lower than for the active ones, but it's significantly higher for the poised ones, for which this has been established. The poised ones are really repressed and become only active during lineage specification. So we thought that's kind of lukewarm support for the priming model. And we went then on to the next prediction that the priming model would make is that these enhancers gain active enhancer marks when cells differentiate and become more specialized. So we've looked at a couple, this is published data. We looked at mesoendodermal cells, trophoblast-like cells, mesenchymal stem cells, and neuroprogenesis cells, and we classified them by whether they would gain H3K27 acetylation in this tissue. You can see that some of them they do in one cell type only. I call them lineage restricted. Some gain in all 4 tissues that we've looked at, but the majority actually don't gain. So we thought, maybe we've looked at the wrong cell types, but this covers quite a lot of directions in which pluripotent stem cells can go. So we thought, what if they are actually, functioning as enhancers already? And for this, we turn to, so StarSeq obviously measures enhancer activity outside of the chromosomal context. So it's important you disturb really the enhancer function, you perturb enhancer function at the endogenous locus. So we turn to an inducible CRISPR-I system. We use the inactively, catalytically dead Cas9 that's fused to PrAT, to repressive domain. This is called CRISPR-I, this system. And you can target that to enhancers. You induce heterochromatin, like a mini heterochromatin domain over the enhancers, and that inactivates their function. And the prediction we have, if they actually work as non-canonical enhancers, as proper enhancers, genuine enhancers in human-induced pluripotent stem cells, you should see a reduction in gene expression. And that's indeed what we see. In some cases, we don't always see it, but in some cases, we see this. I think it goes down quite dramatically, and they're over quite large distances. Yeah, this is, I have a video of this, which I'll try to play after this, because I can't make it play in PowerPoint. But so in one case, we got something much more than we bargained for. And this is this enhancer. When we target that, it's got a nice ataxic signal, clear StarSeq signal. It's also bound by CDCF, interestingly. When we target this enhancer, in addition to the target gene going down, we think is the target gene, we also really see this dramatic effect on cell fitness and cell morphology. And so this is pluripotent stem cells should look like nice colonies. They grow in colonies. When we induce CRISPR-I against this enhancer, we see shell and GFP is just a signal for us that the CRISPR-I has actually been induced. We see the cells look completely different, so they change morphology. A lot of them die. We're trying to find out if they differentiate into something particular. I don't think so. I think it's just they lose pluripotency status. The RNA-seq is just coming back, so we're checking that. But it's targeting this enhancer leads to a loss of pluripotency, and it's an HCK27 acetylation negative, right? So that's it. We still have doubts about this. We still need to make sure that's not an artifact, because it's incredibly quick. We see these effects in the first signs in a day or so. So when Ayub first showed me, I said, it can't be true. It's too quick. You can't inactivate an enhancer and then see an effect on a target gene so quickly. It would require that the RNA and the protein of the gene is extremely unstable and it needs to be turned over so quickly. So I said, no, that's not real. And he did it in another cell type. So we have this panel of 10, took another iPSC cell line, same thing, even more dramatic in that line. So starting to believe it, but we still, still need to check, I think, some things. So we also wanted to, so I'll end on this. We're interested in, and this comes back to what makes these individual lines different, right? These iPSC lines from different donors. And the bigger question behind this is, what makes us different? We looked at regulatory variation in pluripotent stem cell enhancers. We've done StarSeq. I probably forgot to say that, but we didn't clone, which is a key point. We didn't clone candidate enhancer regions for our StarSeq. We cloned the entire genome. So we chopped up the genome. We did genome-wide. So we have this as a genome-wide map. And we have ATAC-seq. We generated ATAC-seq profiles for all 10 of these lines. And most of the peaks are consistent, which is what we want, because they're all the same cell types. But occasionally we find ones that like 3% or so of the peaks that are clearly different. So if you compare this one, and that overlap with the StarSeq signal. So we asked, can we actually correlate this? So we took three of these regions where we do see a StarSeq signal, but the ATAC-seq signal in one donor is much more pronounced than the other one. So the differential chromatin accessibility. And interestingly, that correlates with the gene expression level qualitatively of nearby genes. So I try to keep the same colors here. So where you have a reduced ATAC-seq signal, you have lower gene expression levels. So we ask, if we now clone these sequences, because they're all whole genome sequences, sequence, if we clone them into a star-seq-like assay, in this case, luciferase the readout, but other than that, the assay is very similar. Would this show that this is actually a weaker enhancer than this, and just caused by the sequence variation? And that's indeed what we see. It's quite clear these cases. So whenever there's a stronger ataxic signal in vivo at the endogenous site, actually we can recapitulate that in enhancer reported gene assays and episomal reported gene assays. So these things correlate. Enhancer strengths as measured by these assays, ataxic signal at the endogenous locus, and RNA expression at the locus. And we've now turned to, and this is where I could get in choppy waters talking to people who know a lot more physics than me, and probably about these models, but we're now I'm very excited about this, but it's really in its infancy. We're using a machine learning approach to predict the effect of these enhancer variants, which would be ultimately, our goal would be to understand the logic of enhancers in a way that would allow us to design synthetic enhancers with desired activities. And this is not our model, it's called DeepStar, again by Alex Stark's lab. And this is a comparison, so it's actually doing quite well. So the correlation between the predicted expression and the measured expression is 0.7. It's not perfect, but it's not bad at all. And this is a tracker from this is real data, and this is the DeepStar prediction. And interestingly, then from these, I don't have the data here, but it's just from this morning in lab meeting. But this is the same graph I showed you in the previous slide. It predicts the direction of this effect, predicts correctly in 3 out of 3 cases. You can say there's a 1 in 8 chance it would do this randomly. And it doesn't predict the magnitude as well. It predicts much smaller changes than we see experimentally, but it gets the direction right. So I think it's very encouraging. And my hope is we can build on this and feed the model with better data, that it'll become better, and that reinforces itself. So that's something I'd love to do in the future. And with that, I'll thank. This is my lab, but half of that is my former lab now, or moved on to other adventures. But Young has driven the star-seq experiment that I used on a lot of the CRISPR experiments. This is a collaboration with Ufuk's lab in Göttingen, and Alexander does all the bioinformatics analysis of this, and also with Philippa. I haven't shown the data, but TTC on this. And we have fantastic support at Abraham through our facilities, the genomics facility, bioinformatics, and I haven't put live cell imaging on here, but they've also been great. I'll just before I stop, maybe I can show you this. If I stop to share, wait. I'll try it. If not, you've seen it on the. Hang on. Yeah, it should actually work. Hang on. Yeah, it does work. It doesn't work in PowerPoint, but it does work here. So this is the control CRISPR experiment. Or this is live cell imaging over 4 days, imaged every 2 hours. You can see this is a happy pluripotent stem cell colony. It grows nicely in shape and eventually fuses with others. But this is what you want to see. And then show you this control. Yeah, so this is now where we induce CRISPR against this enhancer, CTCF1 enhancer, shown me previously. So you see GFP comes on, and there's 3 colonies here in the picture. You see they look completely different. They lose their pluripotent morphology and characteristics. Yeah. Okay, and with that, I'll thank you.